Hello, dear students. I welcome you on behalf of Sri Jayendra Rangjong classes. Dear student, we are in a continuation of the chapter that is cell, the unit of life. And in this, in the previous classes, we have already talked about the majority important eukaryotic and prokaryotic difference of these cells. And apart from this, we have also understood about the different mitochondria and chloroplast. But in today's lecture, we are going to discuss ribosome and the cytoskeleton part. So first, we are going to discuss the ribosome. Okay. So if we talk about ribosome, we know that they are the protein synthetic machinery, or they are involved in a synthesis of protein. Okay. And this ribosome was a granular structure first observed by George Belladi. It was first observed by a George Belladi. Okay, and in 1953. So ribosome, if we talk, they are mainly composed of RNA and proteins. So they are having a ribonucleic acid, and there are some proteins in their as their component. And if we talk about ribosome, it is a membraneless organelle. It is not surrounded by any membrane like mitochondria and chloroplast. Okay, so they are membraneless. When we talk about ribosomes, these ribosomes are found in eukaryotic cell as well as prokaryotic cell. But in eukaryotic, they are of 80S type, and in prokaryotic, they are 70S type. Okay, now we remember this 80S ribosome, if we talk specifically, then this 80S ribosome is found in eukaryotic cell, obviously, but in cytoplasm of eukaryotic cell. Whereas they are found in prokaryotic cell, they are 70S, and this 70S also found, so it is also found in mitochondria and chloroplast of eukaryotic cell. Okay, so this is the ribosome. Now ribosome are embedded over the mRNA like this. Suppose this is the mRNA, and over this mRNA you will find this ribosome. Okay, and here when we talk, they are 70S or 80S ribosome. Here S, if we talk, it stands for the sedimentation coefficient, or it is expressed in Swedberg unit, and it is indirectly measure the density and the size of this particular ribosome. Okay, so this is what a Swedberg unit. Now, what is Swedberg unit? First, that we need to understand. So, if you want to understand this Swedberg unit, what Swedberg did, or sedimentation coefficient, he taken a capillary tube, and in this capillary tube they have liquid viscous fluid, and in viscous fluid they have floated this ribosome, isolated ribosome. So the time which they take, okay, that will be noted. Okay, and that time will describe its what size. If it takes a much time, then it is a particularly small particle, but if it takes settle easily, then it has a heavy size. Okay, so according to this we can say the size and the density is particularly understood, or particularly we can say understand. Okay. Now here, if we talk about 1s, one sedimentation coefficient, or one Sudberg unit is 1 into 10 raised to minus 13 second. Means this much time it takes to settle down. If it is a 70s, means 70 into 10 raised to minus 13 second it will take. If it is 80s, then 18 into 10 raised to minus 30 second it will take to settle in this capillary tube. So based on this particular, the sedimentation coefficient is dictated. Now, if we talk, both the ribosome are composed of one larger subunit and one smaller subunit. If we talk about 70S ribosome, or if we talk about the 80S ribosome, they are particularly made up of two subunits. One is larger and one is smaller. See, this is the ribosome, which is 70S, which is found in prokaryote as well as chloroplast mitochondria eukaryote. They have two subunits, 50 and 30S. Now, hope you are wondering that why 50 plus 30 is equal to 80, but it is 70s. Why it is 70s? Because when we take this subunit as a whole, as a 70s, and we float in the capillary tube, it will take a 70 into 10 to minus 30 seconds. But if we take its two subunit and we float them individually in this capillary tube, or we put in the individually in this capillary tube, then it will take a 50 into 10 to minus 13 second for the larger subunit and 30 into 10 to minus 30 second for the smaller subunit. Okay, but combined it will take a less time. So because of this, we are writing it as a 70s. Same happens in the 80s. It is made up of two subunit, that is a 60s subunit and the 40s subunit. Okay, so this is what a particular different ribosomes which are found. Now we can see this ribosome are looking like this. This is the larger subunit, this is which is 50s, this smaller subunit, this is 30s, and the complete is 70s ribosome. Okay, so large subunit, if we talk, they are 60s plus 40s, and they are giving rise to the complete radius ribosome. Okay, so this way, small and large subunits combined and form a complete whole ribosome units. You can see this is the way where larger and subunits fit over each other. Okay, and the complete ribosome form. So this is the structure of eukaryotic ribosome. Okay, and if it is a structure of eukaryotic ribosome, you can understand that this is the 80s ribosome, and obvious 80 has a larger subunit, that is 60s, and smaller subunit which is 40s. Okay, so this is the thing which is found here. Now the two subunit in both type of ribosome associated with each other in the presence of magnesium. Means magnesium is required for their association. When certain amount of magnesium is there in the cytoplasm, then only they are going to associate with each other. And ribosome provide a space as well as enzyme for the synthesis of protein. Means because of this we call it as a protosynthetic machinery, because they are particularly providing a space as well as we can say a synthesis of proteins. Okay, now if we understand this large and small subunit, they have a RNA and we can say the ribosome. So some more important points we are going to note down here. This is membraneless organelle. We have studied already some microscopic. Okay. See, it was, we can say, first observed by Pallade by electron microscopy. So Pallade observed ribosome in animal cell. But in plant cell, if we talk, Robinson and Brown Robinson and Brown first noted ribosome. First noted ribosome in plant cell. Okay. Now, if we talk about its occurrence, ribosome occur in all cell, and we have studied the cytosolic ribosome are ATS and organelle ribosome are 70S. They have two subunits, that all things we have noted. Now, if we talk about a 70S ribosome and their component, suppose this is a 70S ribosome, then this is a 50S subunit, and this is the 30S subunit. So the 50th subunit if we talk and 30th subunit, that is 70th ribosome forming, and they are obviously we can say the ribosome, and they are obviously rRNA and protein complex. Now if we talk about rRNA which are present in 50th subunit, then they are 23S rRNA and 5S rRNA, and in small subunit they have 16S rRNA. Apart from this small subunit, a large subunit, if we talk, that is 50S, they have 34 protein molecules. Whereas this small subunit, if we talk, which is 30S, has a 21 protein molecule. So this much thing, these are the type of rRNA which are found, and these are the number of proteins which are found. And in 70S ribosome, if you do, they are the ribonucleoproteins because they have a nucleic acid as well as proteins. So they have a six. They have, if we take 70S, 60 is to 40 ratio of RNA and protein. Okay, but if you talk about 80S ribosome, they have a larger subunit and they have a smaller subunit. In larger subunit, you will have a 20S rRNA, then you will have a 5S rRNA and 5.8S rRNA, and apart from this, you will have a 18S rRNA in this subunit. This is 40S subunit, this is 60S subunit, this is 80S ribosome. Okay, now here if we talk, they have some additional proteins. Then in larger subunit you will see that there is a 40 protein molecules are found, and in 40A subunit if you talk, 33 protein molecules are found. Okay, and in 80S ribosome you will see the RNA is to protein ratio, that is 40 is to 60. RNA is to protein ratio. So this is the what constitute composition of the this A70 and 80S ribosome. Okay, okay. So this is what about the ribosome. Now, if we talk about the origin of ribosome, then in origin, it is known that in prokaryote, both RNA and proteins both RNA and proteins are synthesized in cytoplasm in eukaryotes. Okay, so this prokaryotes, both RNA and protein, are synthesized in the cytoplasm of eukaryotes. Okay, so in eukaryotes, this both are RNA are synthesized in the cytoplasm specifically. Specifically, if we talk about the rRNA, this rRNA are synthesized in nucleolus. Actually, remember this rRNA. This is different. Other RNA can be found in the cytoplasm, but usually, if you talk about the RNA, it is found in the nucleus. So I'm removing this. Okay. not both RNA and prokaryote. In prokaryote, okay, sorry, yes, you can write in prokaryote RNA and RNA, just a minute, RNA and proteins are synthesized in cytoplasm, whereas in eukaryote rRNA are synthesized in the nucleolus of nucleus. Okay, so we remember this is the occurrence or origin where they are found. Now, if we talk about the function of this ribosome, dear student, you will understand that the free ribosome, free ribosome synthesize non-secretory protein. Okay, non-secretory protein means the proteins which are synthesized for the use of cell, for itself a cell. It is not going to secrete it out. That are going to used within the cell. So that type of proteins are synthesized by the free ribosomes. Okay, but if we talk about other ribosomes that are ER bound ribosomes, so bound ribosome synthesize secretory proteins. Okay, and it is known that the after a so protein synthesis is a translation process. Okay, so proper folding of protein after its synthesis is done by chaperones protein. Okay, so this chaperone work after the translation means a process of protein synthesis. After translation they are going to work, and after translation when they work they carry out post translational modification, and this post translational modification proper folding of this protein is going to take place, but this is carried out by the this is carried out by the chaperone protein. Okay, so this is what about the ribosome. Now we are after discussion of ribosome, we are going to talk about the cytoskeleton. So let's talk about the cytoskeleton part. Now we know that in our body, if we talk about the human body, we have a skeleton system, bones. Because of this, our body has a one posture, one standing position, or our body has a different organ and their particular structure. Now, same way within a cell, if you talk, they will have a such skeletal structures, and these skeletal structures are known as what we can say a cytoplasmic skeletal structure. They are the cytoskeleton. Now, in this, three components are mainly used here. Okay, so in cytoskeleton, if we talk, then it is an elaborate network of filamentous proteinaceous structures. There are what? Filamentous means long proteinaceous structures are there which are present in the cytoplasm, and they collectively referred as a cytoskeleton part. Okay. Now, eukaryotic skeletal cytoskeletal structure contain, if we talk about this, then this eukaryotic cytoskeleton structure contain three components. One is microfilament, another one is the microtubule, or intermediate filament, and the third one is the microtubule. Okay, these three are the particularly proteinaceous structure, filamentous structures, which support the extensive system of membranes within the what eukaryotic cell, and they perform a various function, and they are collectively termed as a cytoskeleton. They collectively termed as a cytoskeleton. Okay, so in this, if we talk about the first, that is the microfilament. Okay, then this microfilament are the filamentous structure which look like this. Okay, and this microfilament have some actin proteins, and particularly this are helping in formation of this. Now if we talk about this microfilament, microfilament, then dear student, these microfilament are obviously found in eukaryotic cell, and these microfilament are solid, unbranched. They are unbranched. Okay, so solid hai, they are solid, they are unbranched, they are rod like fibrils, they are rod like fibrils of indefinite length. They are of independent length, and it is known that they are mainly composed of globular protein that is actin. They are mainly composed of globular protein that is actin. Okay, but have filamentous protein myosin also. Okay, and they form an extensive network in the cytoplasm of cell, and they may be found associated with plasma membrane. Okay, so this is the what is microfilament. They are solid, they are some solid component like this, and they are long filamentous structure of indefinite length, and they are made up of actin and myosin proteins. Such long filamentous structure are found. Now we have to understand what is the function of them. So if we talk about the function, the function of microfilament, if we talk, first function is it support to plasma membrane. Second is this microfilament represent contractile system of cell, and involved in cytoplasmic streaming or cyclosis and amoeboid movement. Okay, so the contractile system means they are mainly involved in contraction or relaxation, and they particularly helping in the cell that a particular component of all the cytoplasm keep moving in a particular manner. Okay, if we talk about another function, then it also help in spodopodia formation, and fourth is the help in formation of cleavage furrow during cell division in animals. Okay, so these are the functions which are carried out by the microfilament. So let's continue about the next one, that is microtubule. If we talk about microtubule, dear students, these are also filamentous proteinaceous tubular structures. Okay, so if we talk about microtubule, so if we talk about this microtubule, these are found in cytoplasmic matrix of a eukaryotic cell. So if we talk about this microtubule, they are hollow, unbranched cylinders. So they are hollow, unbranched cylinders. And if we talk about this hollow unbranched cylinders, they are generally 25 nanometer in diameter with a hollow core of 15 nanometer. So suppose this is the cylindrical and this is the cylinder like this and this is the top part. So this having a what a 25 nanometer, but the central cylinder is a 15 nanometer. Okay, so such a way these are found. Okay, so now this is generally have a 25 nanometer diameter with a hollow core of 15 nanometer. So in this, if we talk about this boundary, okay, so this boundary, if we talk, they are made up of, okay, so if we want to draw, suppose this is the microtubule, and if we talk about this microtubule, if we draw in a broader way, then so here total 13 protofilament are found. Okay, so here 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13. 13 protofilament are found. Okay, in the periphery part, in this part. Okay. So 13 protofilament are there. Now in this, each protofilament, if we talk, each protofilament is made up of alpha and beta subunit. Each protofilament is made up of alpha and beta subunit of tubulin protein. Okay. Now it is known that the assembly and disassembly of microtubule, if you want to associate this type of microtubule, so many microtubule, then the assembly and disassembly of microtubule require GTP and calcium. Okay, so GTP and calcium are required for the assembly and disassembly. So this is the what a particular microtubule. Now where they are found. So it is known that They occur in microtubule occur in cilia, flagella, centriole, and basal bodies and mitotic apparatus. Okay, so in this they are found. Now, if we talk about the functions, then function of microtubule include number one, help in spindle and astral ray formation during cell division. So they help in spindle and astral ray formation during a cell division. Second is they form a cytoskeleton of cilia and flagella. Okay, so astral ray and spindles, which are going to be used in a cell division, they are formed during cell division by this one. They form a cytoskeleton of the cilia and flagella, flagella. So by this, when we talk about the third function, they help in they help in generating the shape, rigidity, and form of cell and cell mobility. This all are particularly maintained by this microtubule. Then if we talk about the fourth function, then microtubule help in anaphasic movement of chromosome. So during anaphase, the movement of chromosome which occur, that is due to this, we can say a microtubule. And then fifth function if we talk, that is microtubule help in intracellular transport. Intracellular transport means transport within the cell of nutrients and inorganic ions. Whereas if you talk about the sixth one, then the sixth function is position of future cell plate during cell division in plants. Okay, so these all functions are carried out by the microtubule, which are also proteinaceous filamentous structures. And the third component of this cytoskeleton part, which I'm going to discuss, that is a intermediate filament. Now, see, intermediate filament, if we talk, they are not much more prominent, but it is known that if we talk about intermediate filament, these are a hollow filament of acidic protein, and they are involved in formation of scaffolds for chromatin. Scaffolds for chromatin, and in forming basket around nucleus. Now we remember scaffold for chromatin means, suppose this is a nucleus, and in this nucleus they are forming a basket, means they are present like this. So this is the nucleus, then they are forming, and surrounding to the nucleus they are arranged like this, so that form basket. And if they are here, the chromatin material is there, somewhat chromatin material is added here and somewhat is added here, means they are providing hook like structure to this chromatin material, so that are known as scaffold. So this is a chromatin and this is a nucleus, and this is intermediate filament. So such a way they help here in the particular this structure. Okay, so that is about the what cytoskeletal structures. Okay, so cytoskeleton functions we have already discussed so many. So mechanical support they can provide, maintenance of cell shape, cell mobility, intercellular transport, signaling across the cell, karyokinase, all they can help. So if you talk about some MCQs and easiest MCQs, all cell organelles are membrane bound except. So lysosome is single membrane bound, glycosome is also single membrane bound, nucleus is also single membrane, ribosome one membraneless obvious. Then the cell organelle present in both prokaryote and eukaryote. In both prokaryote and eukaryote, you will find a 70s ribosome, because in mitochondria of eukaryote you will find a, and you chloroplast of eukaryote and mitochondria you can also find a 70s ribosome. Now proteins in the plants there are some. So proteins in a plant cell are synthesized by. So obviously protein synthetic machinery are ribosomes, so ribosomes are involved in this. Association and dissociation of ribosome are mediated. Obvious, this is mediated by the magnesium ion. We have studied that. Cytoskeleton is produced from. It is produced. Cytoskeleton is the produced from. So it is made by the microtubule and microfilament. Okay, so that is all about the ribosome and the cytoskeleton part. I hope you understood. Now, in a continuation of the cytochrome, the structure of cilia and flagella we will understand in the next lecture. Okay, thank you.